Experiment number 4. After this lab, students should be able to observe stages of embryonic development in frog and also observe the embryonic development in chick. Embryonic development in frog. Frog have been used widely in this animal model as there is of observation from the fertilized egg to a tadpole stage. Tapaka frog is a runner penguin, is developed at 80 degrees. This life cycle takes from 7 stages, which is from unfertilized egg to a tadpole, which is around 12 days to complete. Materials needed for this experiment. This is a stages of development of frog, from undivided, early cleavage, late cleavage, blastula, gastrula, neurula, and tadpole. Here is the pictures of these development stages. From microscope view, we can see that this is undivided fertilized egg, which observe the egg yolk adjacent to the cytoplasm. Second is the early cleavage. A fertilized egg was first divided into 2 cells, then later to be 4. At these 2 stages, each cell will be called blastomere. At 4th cell, blastomere cell will be different in size. At the late cleavage, at this stage, the embryo has more than 4 cells and the blastocele begin to develop. The next one is blastula. The embryo will form a cavity that we call a blastocele. The next one is gastrula. Gastrula form when they increase in size form a cavity that we call a blastopore. At this stage, 3 layers of embryonic tissues will form that we call is mesoderm, ectoderm, and endoderm. Blastocele reduce in size while the gastrocele become bigger in size. Next is neurula. Embryo increase in length. At the dorsal, there is a neural plate. The head and the body can be differentiated. And the last one is a tadpole. The brain cavity is formed and look at the pharynx, notochord, liver, and also yolk. This is they have external and internal gills. B. In embryonic development in chick. Normally, chick develop on land with this. There is no larvae stage. Therefore, chick development is markedly different from the frog because of these 3 features. Number 1. Embryo will be enclosed by the extra-embryonic membranes. Number 2. There is a large amount of yolk to sustain development of the chick. Number 3. There is a hard outer shell. Material needed is chicken egg, petri dish, and chick animal model. Here is the day 1 of the yolk, which have a blastodisc with the nucleus. Day 3, day 4, day 5, and after 21 days, the chicken will be hatching from the egg. that's all from us. Thank you very much. Good luck and assalamu alaikum warahmatullahi wabarakatuh.